first is the introduction, why we want to use nanotechnology for stroke treatment. And the second part will be the proof of concept, how we're going to using the rotating magnetic nanomotors to enhance the efficacy of the stroke treatment. Then the last one, essentially, we're going to achieve animal models that can do both the targeted delivery of the drug as well as enhance the stroke treatment. The last session will be the conclusion and also the future perspective of this technology. So essentially, what is a stroke? Actually, according to the World Health Organization, stroke is the #1 leading cause of disability of people in the old age, and also #5 of the cause of death from all these diseases. So every year, about 15 million of people actually worldwide suffer from stroke, and then 5 million of them died from the stroke. According to US statistics, so every year there's about 140,000 of US died about stroke. That means it is about 1 out of every 20 deaths, or every 40 seconds there's somebody in US actually suffer from stroke, and then every 4 minutes there's somebody died due to the stroke. Obviously, this kind of rate is higher in China or other countries as well. There's two kinds of strokes. One is called hemorrhagic stroke, which is essentially you have the internal bleeding in your blood in the brain. Another one that is actually account for 15% of the stroke suffering. When you have this hemorrhage strokes, usually people have to go to the emergency room. Another one is called this ischemic stroke. Usually that causes a lot of disability, which is causing by blood clot actually block the flow of the blood inside the brain. That counts for the 85% of the stroke. So obviously, we are mostly concerned on this 85% of stroke patients due to that kind of high percentage of patients. There's two kinds of methods to treat the stroke. One is that, mechanical remove by surgical. So if you have large blood vessels that blocked by the clot, you can use the mechanical stent actually mechanically remove the stroke. However, if the stroke happened in the brain because of the blood vessels are really small, then there's only one way to do that. Essentially, you have to use drug to treat it. However, there's only one approved drug by FDA that is called tissue plasminogen activator. So you can infuse this drug into your blood, and once the drug actually reaches the stroke site, it will dissolve the blood clot. Now, the tissue plasminogen activator essentially is an enzyme. The activity, how it will dissolve this blood clot, essentially is complicated. There's one activation path. Also, there's a multiple inhibition path. So this is actually an image of the blood clot. You can see there's a lot of fibers connected with blood clot, red blood cells, and also platelet, and also many other small particles. So when you have a tPA injected into the blood, it will actually react with plasminogen actually to activate this plasminogen into the plasmin. This plasmin actually will react with these fibers, which is called fibrin fibers, that actually form this blood clot. So it actually will dissolve this. So after multiple times of reaction, then the entire fiber structure will be destructed, and then the whole blood clot actually can be dissolved. And this drug has to be treated within the 3 hours or 5 hours of when you find you have a stroke in your brain. Now, unfortunately, there's a big side effect of this tPA treatment. If you have a high concentration of tPA, this tPA always causes this so-called symptomatic intracranial hemorrhage, which actually causes the internal bleeding of the blood vessel in your brain. So people find that there's about 6 to 7% of the patient treated with tPA has such a effect. And also due to the small size of the blood clot, blood vessel in the brain, so usually we have to using different method to enhance this kind of treatment and also to eliminate the size effect. There's 3 different ways people thinking about to do that. First one is that to do the dose control. So if we can actually minimize the amount of tPA while it's circulating in the body, then obviously it will have less chance to cause the internal hemorrhage of the blood vessels. And also, we should also improve the efficacy of the blood clot treatment, so we can enhance the reaction rate around the blood clot that actually can improve this treatment. Obviously, to do that, we want to do so-called targeted delivery. So all this dose control and targeted delivery are all the purpose of so-called nano drug carrier research that has been also being used for cancer treatment, tumor treatment, and others treatment as well. Now, if we look at how much tPA get administered in our body, usually the amount of tPA is very small. It's less than 100 micron per milliliter. So we did a study about if we change the concentration of the tPA, how would the blood clot lysis rate? Actually, in our experiment 8 years ago, we found that the blood clot lysis rate as a function of the concentration of tPA essentially follow this curve. So at a very low concentration, you can see that clot removing rate is almost linearly increased with the concentration, but when the tPA concentration is larger than 200 microgram per milliliter, the rate actually is level out. So essentially it's a constant. So in this low concentration region, if the lysis rate is proportional to the concentration, that means the reaction is limited by the transportation of the drug. So in chemistry, this is always called diffusion limited reaction. So according to diffusion limited reaction, the reaction rate is proportional to the concentration of the drug, also is proportional to the diffusion coefficient of that drug at that location, and also is proportional to the size of the molecular and also the reactants as well. So according to these principles, there's 2 ways we can increase the reaction rate. One, obviously, is increase the local concentration of the TP around the blood clot so that the essentially reaction concentration of tPA is higher, while the transportation concentration of tPA is low. Another one is obviously to increase the mass transport of the tPA around the local area of the blood clot. So essentially, that will enhance the mass transport. Unfortunately, for most of the drug carrier kind of research, people are concentrated on to increase the local concentration of the drug around the disease area. However, to enhance the diffusion coefficient or mass transport, nobody actually have done before. So based on this, in the literature, there's 2 kind of general catalog of nanotechnology for stroke treatment. We catalog them into so-called passive nanoparticle drug delivery. So essentially, you have particles and then incorporating different kind of drug into the particle, and then you inject the particle in your blood system, and then hopefully those particles will targetedly treat those disease area. Another part is the so-called active nanoparticle treat delivery. So when the particles are concentrated around the blood clot area, people can using either ultrasound or rotating magnetic field or the light to activate the motion of the particle. And then using the motion of the particle and also the hydrodynamic principle, people can enhance the mass transport of the drug released from the particle, so we can enhance the reaction rate. And to do this nanotechnology-based stroke treatment, there's basically 3 steps. First, we actually, from the material science point of view, we need to load the drug onto the nanoparticle. And then second, in order to achieve the so-called targeted delivery, we usually put down some functional ligand that can specifically target the blood clot onto the surface of those drug-loaded nanoparticle. And then once it transports through the circulation system and reach the blood clot location, then we can using some stimulate mechanism to release the drug locally. There's 2 different kinds of stimulated mechanism. One is caused by physical interaction. One is caused by chemical stimulation. For example, one can use pressure to break the particle. One can actually use the motion to improve the release of the drug. One can also use the heat to help dissolve the drug from the surface of those drug-loaded particles. One could also using the chemical environment, for example, the change of pH value, the generation of so-called reactive oxygen species, or using the light dissociate the covalent bonding into the particle and the surface of the nanoparticle, or using specific molecules to cleave the drug actually bonded on the surface. So this entire process is complicated. And so based on this, here we are going to talk about how to using the enhanced mass transport to improve the stroke treatment efficacy. So the idea is very simple. If you can actually deliver nanorod, magnetic nanorod in the stroke location where you have the blood clot, then if you actually putting a rotating magnetic field to make the particle rotate, I mean, this is just like a small mini magnetic stir bar that can actually help the improvement of the chemical reaction. So you can see that, okay, in this simulation, these green particles are drug molecules. So essentially, initially, they actually are diffusing into the blood clot in the microchannel according to this Stroke-Einstein relationship. However, when the particle actually get bounced by this rotating magnetic nanorod, their relative diffusion rate actually can be increased. So according to the hydrodynamics theory, the effective diffusion coefficient of those molecules inside a rotating magnetic rod actually will depend on how fast the particle is rotating and also what is the size of the particle. So according to that, the effective diffusion coefficient is proportional to 1 plus 2 over 3 pi squared over PE, which PE is called the Planck number, which P is proportional to the rotation speed of the nanorod and also the overall dimension of this nanorod. So that means if we introduce the nanorod inside the clot and then rotate them, then essentially we should be able to increase the drug treatment efficacy. So in order to demonstrate that principle, we first making some nickel nanorod based on a technique called oblique angle deposition. So on a glass substrate, we first put it on a monolayers of polystyrene beads. Those beads are actually etched into a very small size. And then we put the whole thing into a thermal evaporation chamber while we can evaporate nickel material at a very large angle with respect to the surface normal of the substrate. So when the vapor comes at a large angle, there's so-called a shadowing effect. That shadowing effect will ensure that all the vapor will only come to the nano beads, polystyrene beads. So here is a cross-section image of this nickel nanorod on this polystyrene nanorod, nanosphere bead. You can see each bead actually have a rod over here. So once we making those substrates, then we can actually sonicate those particles off from the substrate into the solution. So once they sonicated off, okay, they become individual particle. Here is a SEM picture of this individual particle. you can see the overall length of the particle is around 1 micron. The diameter of the particle is about 2 to 300 nanometer. And here essentially is a suspension of the nickel nanorod in the liquid. So with this particle, we can actually design a microfluidic experiment. We actually have 8 channels. At the end of these 8 channels of the microfluidic, at the end, we actually put down blood clot. This essentially is fibrin networks on the end. It does not contain red blood cell, but it's a fiber network. So in this channel, you can see that we designed a setup. So this part at the end is the blood clot. In the middle over here, we put down a dye solution. We have a color, so we can using computer to differentiate the interface between the liquid and also the blood clot. And then at this end, we actually put down oils to prevent the evaporation of this liquid over here. So this dye liquid over here, we actually put different kind of ingredients. First, we only put down like the dye over here. We only put dye over here. We also in one channel we put only the magnetic nanorod over here. In one channel we put the dye plus tPA over there, and in another channel we put a mixture of nanorod with a tPA solution together. So we can do this 8-channel experiment simultaneously into in-plane rotating magnetic field, and then we can use a microscope actually to take a picture and then form a movie of the whole system. So here shows a movie of how the treatment looks like. Let's see. Somehow, the movie does not show up over here, and
yeah, Professor Zhao, it may be so if you can off the laser.
Okay. Anyway, so based on all those 4 channels, what we get over here is that we can using the computer to identify the surface at the interface. For the dye-only surface and also the magnetic motor solution surface interface of channel over there, we can see that, okay, with the increase of the time, it shows a typical diffusion curves that essentially give us the diffusion of the dye solution into the blood clot network. However, at the tPA interface, we can see that the interface increases, okay, as a function of time. And at the nanomotor with the tPA with the rotating magnetic field, the interface actually move faster. So based on this graph, we can actually derive the blood clot lysis speed as a function of the concentration of the of the nanorod in the solution with a fixed tPA concentration. So this figure shows the speed of the lysis of the blood clot as a function of the nanorod concentration. So the red dot is the rod with the same concentration of tPA, and then the blue dot actually is with the same concentration of tPA. So you can see that with the same concentration of tPA, the lysis rate is almost a constant. However, with the increase of the concentration of the nanorod, it increases monotonically with the concentration. Based on the equation previously I showed, we found that the lysis rate should be actually proportional to the concentration of the rod to the 2/3 of the power. So this curve over here, the dashed curve over here, is a fitting for the experiment data that actually fits very well. Obviously, this is from the principal point of view, how the mass transport will help to increase the tPA, the drug treatment. And we also did a very simple mouse experiment. We have two separate experiments. One is that first we have do a surgery of the mouse so we can induce blood clot in this blood vessel of this mouse. So we treat this mouse with three different solutions. One is a control group. We only treat the mouse with PBS, the buffer solution. Another group, we only treated with tPA without any magnetic nanorod. The third group, we treat with both the tPA and the nanorod with the rotation. So essentially, the mouse is fainted over here and then put it in this magnetic setup, and then the magnet actually rotating 24 hours. So after 24 hours, here is the result. For the control group with the PBS, since there's no drug, so the blood clot essentially still remain over here. The black one over here, black section essentially is the blood clot. And for the tPA treated only group, after 24 hours, there's still some blood clot remained in the blood vessel. However, for the tPA plus the nanorod treated by a rotating magnetic field, after 24 hours, the blood clot actually is gone in this graph shown over here. So this experiment demonstrates the principle of how to using the mass transport to enhance the treatment of the blood clot. Then obviously, the goal is that we want to achieve these three kinds of treatment. One is a dose control, one is a target treatment, target delivery, another one is efficiency improvement. So in order to do that, we cannot just mix the drug together with the nanorod. Essentially, we can load the drug onto the magnetic nanorod through a very simple chemical surface reaction. We can load the drug onto the surface of the nanorod, and also we're using iron oxide, essentially it's magnetized nanorod. This is more biocompatible compared to the nickel nanorod. We found that, with the rotating magnetic field and without the rotating magnetic field, the drug released from the surface of those magnetic nanorod are different. So without the rotating magnetic field, the drug will release due to the physical desorption of the surface. But with the rotation, you can see the drug release rate is much higher. It's like almost 50% of improvement. So that means we can use in the rotating magnetic field also to control the drug release as well. So we actually did a bulk synthesis of magnetic nanoparticles using the so-called hydrothermal method. We first making those iron 203 magnetic nanorods, and then we actually oxidize those iron 203 nanorod into iron 304 nanorod by oxidation. And here is the TEM pictures of how this nanorod looks like. And essentially, those nanorods are highly porous. You can see that there's multiple small particles actually aggregate together into a sub-micron size of particle. If we look at the high resolution TEM, essentially the single particle essentially is smaller than 10 nanometers, right? So you can incorporate a lot of drug into those porous nanoparticles. Here is the XRD to confirm that the particle actually are magnetized, and their magnetic property shows that they are super magnetic. Okay, so, and also here is a figure shows the loading of the drug onto those surface, and the activity of this tPA actually remain about like 75% of active of those particles. The mass loading ratio is about 13% in those drugs. So based on those particles, we actually did in situ blood clot treatment in my collaborator's lab. So essentially, they induce blood clot in the brain of the mouse, and then they inject the tPA or the tPA drug-loaded magnetic nanorod into the vein of this mouse, and then we treat the mouse with a static magnetic, first to concentrate the magnetic particles around the stroke area, and then later for 5 minutes, we actually rotate the magnetic field to do the treatment. So here shows the result. So in the first panel over here, this is the healthy, blood over here, blood vessel. And then in the second panel, we introduce a blood clot at this location, this location, this location, this location. Now in the row, actually we did 4 different treatments. For the first row, we inject the normal saline, essentially it's PBS, there's no drug. And the second row, we inject 10 microgram per milliliter of tPA into the mouse. In the third one, we only inject 1 microgram per milliliter of magnetic nanorod. And the last row, we inject 1 microgram per milliliter of tPA-loaded magnetic nanorod, and then treat it with rotating magnetic field. So you can see that in the third row, for this first panel, first row and the third panel over here, with the normal saline, there's no treatment effect after 60 minutes. Even after 120 minutes, obviously, the blood clots still remain on the, in the mouse. However, for the tPA side, after 60 minutes, you can see, the blood clot gets dissolved a little bit. Then after 85 minutes, it's totally gone. For this magnetic nanorod, only treatment after 5 minutes, because we're using a static magnet to concentrate the particle around blood clot location, you can see there's a concentration of the magnetic particle around the blood clot location, right? So even after 102 hours, the amount of magnetic particle actually is decreased. So they actually can penetrate into the blood clot location, but the blood clots still maintain over there. However, for this 1 micron tPA-loaded drug, after 5 minutes, we concentrate the particle around blood clot location. Then after 24 minutes, you can see that the particle, magnetic particle, actually go to the other side of this blood clot. And essentially, the blood clot actually is removed, right? So if you look at the efficacy over here is that, so for the tPA treatment with 10 microgram, it takes 85 minutes. However, for the tPA-loaded drug, it only takes like 24 minutes. So that is about 1/3 of the time. However, the amount of tPA actually treated using the tPA-loaded magnetic rod, remember that we only load 13%, right? So that is 13% of 1 microgram. That is about 0.13 microgram compared to 10 microgram. That is like we have 2 orders magnitude of less drugs actually injected into the animal, and then we get like the lysis speed is 3 times faster. And also we look at the toxicity of the treatment by looking at some of the biomarkers in liver and also in kidney. Compared with the experiment treated by normal saline, you can see that the level of all those biomarkers are within the fluctuation of the normal treatment. So they don't have any biotoxicity effect. We also look at the slides of different organs in the mouse treated by different magnetic particle and also salines. So here is the control sample. You can see there's low particles. So after 1 hour's treatment of tPA-loaded drug, you can see, in liver, spleen, and kidney, and lung, and brain, they have some particles showing up, right? And then after 24 hours, the amount of particle actually gets less and less. So if we do a statistic on the amount of particles, you can see, 1 hour, you have a lot of particles actually concentrated in liver and spleen. And after 24 hours, the amount of particles are significantly reduced. So where does those particles go? We actually collect the urine of the mouse after 24 hours. So in the urine, we can see that there's many magnetic particles. So essentially, all these particles will get passed through the body of the animal and then go to the urine. So this method demonstrates that we can, using the static magnetic field, to concentrate the particle to the blood clot location, and then using the rotating magnetic field to enhance the treatment of the blood clot. So future perspective of this technology we are thinking about is essentially what we can do is that we can first do a MRI, three-dimensional MRI image of the blood clot location, and then locate the location, and then do a computer interface so we can design a magnetic hermit. Then we can concentrate the magnetic field around the blood clot location. So when we inject the tPA-loaded magnetic nanoparticles into our body, then we can actually, using the computer system, to concentrate the particle around the blood clot location and then apply a rotating magnetic field about that. Obviously, in order to achieve that, there's still a long way to go. So here, a conclusion is that we can show that magnetic nanomotor with the drug loaded, we can achieve both the dose control, efficiency, promotion, and also the target delivery. And there's still a lot of challenges in terms of nanoparticle synthesis, transport of the particle in the complex circulation system, particle clot interaction, and also magnetic hermit design for the commercial application. With that, I would like to thank you, my collaborators, and also all my students, and also the support from